as you said, it is like maybe a sort of exam oriented thing, or maybe some short questions have been asked about this future vaccines. So it made me read so much, and I think this is need of an hour. And future vaccine we should be knowing, that is for sure. It is not that just the people who are going exam going should be knowing. But so what I would like to touch upon is the basics of all the vaccines, vaccinations, and then we'll be talking about the future vaccines, and that way we'll go ahead. So first, anybody can ask me that what are the how many vaccines are in the pipeline. So my answer can be, it is not an over exaggeration to say that there are uncountable number of vaccine candidates on pipeline for its approval to enter in preclinical trials. Almost every living pathogen which can harm the human, animals, or even fishes becomes the potential candidate against which the scientists are in search of some preventive mechanism, including vaccines. So you name it, name the thing, and scientists they have them. So not only living organisms, even vaccines are also being developed against many chronic diseases like systemic sclerosis, multiple sclerosis, melanoma, and other cancers, and many more. So what are the vaccine development stage that we have to understand? And this is very basic, and we all should be knowing it, that because the whole talk will be divided, like based on these particular things, and every time we'll be using these words somewhere in here and there. So what we are doing it, how the vaccine development stages are coming up, that is to be understood. First is the exploratory phase. There the scientists they try to find out a natural or synthetic antigen. That is exploratory phase, that lasts usually for 2 to 3 years. Then comes the preclinical stage, then clinical development, then approval stage, and then manufacturing process. Now what happens in COVID scenario, we have these many stages we are clubbing together for the fast track vaccination. So but the basic steps for any new vaccine to come into market, at least they have to pass through all these stages, and it will be taking almost 5 to 15 years. Now, as I said, exploratory phase, the basic laboratory research by scientists to identify the natural or synthetic antigens. Once they find the antigen, then the preclinical stage comes. There we would like to see how this vaccine will work. Like usually you look at a several different vaccine candidates, and ultimately you choose one of them, and that has to be giving the best immune response and evidence of protection. So here we will be talking about the animal studies. So we will be studying on the animals like mice and monkeys to see if it produces the proper immune response. Now, is this stage particularly we would like to see the safety and way to administer vaccine and other immunogenicity somewhat. So this is particular stage which is done on a animal studies. But yes, also scientists can do the challenge studies. Now, what is challenge study? You give the vaccine to the animal, and then you give the infection to the animal, and then you look at the response, whether that animal is surviving that infection or not, whether that vaccine has been effective or not. That has also been part of it, this preclinical stage. Then comes the clinical development. Now in clinical development, there are stage 1, 2, 3, and 4. Now phase 1, we would like to see, is this vaccine safe and what is the right dose? So phase 1 is testing, it marks the first time the vaccine is tested in a small group of adults, maybe around in 10, 20, 20 to 40. So it is there to evaluate the safety and measure the immune response in general. That is, it generates. Now phase 2, we go from few subjects to hundreds of subjects, maybe 100 to 500. And here again, we would be definitely looking at the safety and the dose, the immune response. But simultaneously, we'll be looking at the different dosages according to age. What is the schedule? We can say how many doses and method of administering vaccine. These all to be seen in stage phase 2. Here, studies are randomized and placebo controlled both. And phase 3, that is very important phase, because there is we will be measuring the efficacy, that how effective is the vaccine. So in this stage of clinical trial, even more volunteers, maybe thousands of people will be enrolled, and then it will be studied for the outcome. So these usually last for 3 to 6 months, and this is very, very important phase in the vaccine development, because after completion of these phase 3 trial, you get for the approval or licensing. So then comes stage phase 4. So when we just talk about a vaccine different stages, this is a regulatory approval licensure stage, where what happens, is the vaccine ready for the world? That is the question. So it is typically expected that there has to be at least 2 successful phase 3 trial, which is demonstrating the drug safety and efficacy in order to obtain the approval from appropriate regulatory authority like the FDA or European Medical Association. Now, after successful trial, phase 3 trial, vaccine manufacturers submit their application and finally they get the approval. So now, if you think like if you get a vaccine in a phase 2, we are done? No. From preclinical to the final product, it is only 7% success chances. And those vaccines who are in phase 2, they only 20% they reach to further stage 3. And even after phase 3 study, only 30% of the study vaccine they reach to the final market uh, proposition. So this is a development stage, and yes, because of the COVID situation, because there is a huge demand and there is no treatment for COVID, we have a fast track trials. Now, what is that? That we combine 1 and 2 phase trial together and phase 2 and phase 3 trials together. So entire process of developing this vaccine from preclinical research to marketing can be approximately 12 to 18 years. And usually it is a huge cost. But here we just try to decrease that cost, the duration of this vaccine manufacturing. Ebola was till now the fastest vaccine manufactured. That was within 5 years, and I think we are now heading towards making a record that COVID vaccine we may be almost preparing within 12 to 24 months. So to fast track this, we may use the human challenge studies. As I said, that human challenge studies are there in the animal model. Similarly, the persons, they're just what we did in the typhoid, type bar TCB, they did the human challenge study later on. But what they did, they selected the adults who had the naive of typhoid, and then they gave the vaccination, and then they gave the infection with the typhoid orally. And then they tried to see how many of them they developed the fever and then followed by the culture. So that was a human challenge study. The people are trying for human challenge study in this COVID also. So for the fast track trials. Let us see, because WHO has still not approved that. There are almost enrollment has been done in thousands of people. They are ready to go for this kind of trial human challenge studies. So that is what I told. I talked about that human Oxford TCV human challenge trial is now well known, and researchers are exploring this particular in the same thing. Now, what are the cautions in fast track trial? Because vaccine trial may take months to year to complete, since sufficient time period has to be there for the subject to react to the vaccine and develop the required antibodies, because then we can say it has been protected. But what happened in 1955 when John Salk first invented that vaccine IPV? That vaccine was licensed within 2 hours of its announcement, and it was available for the general public. And unfortunately, there is a famous Cutter incident where there was some manufacturing issue with the Cutter company, that contracting the virulent strain of the polio virus, they caused the paralysis. And ultimately, that IPV, which was a good vaccine, which came ahead of OPV, was then stopped there. The studies were stopped, and then it took a long time to come again into the market. So vaccine trials and manufacturing must be handled with extreme caution to avoid the infecting recipients or the causing adverse effect. So that is a caution about fast track trials. Now, as I said, that the type of, and so there was one more thing, that is a phase 4 trial. The phase 4 is like vaccine has been licensed, it has been marketed, but still the thing is not over. Company will be going on, the ongoing research will be there. What are the effects? What are the side effects? What is the happening immunogenicity? These trials are going on, because even in phase 4 trial, you might find some complication like Rota Shield. It was marketed, they were almost given for 1 year, and for phase 4 trial picked up the side effect of intussusception. In those patients who were given Rota Shield, almost 1 in 10, that was the incidence of intussusception. That's why this vaccine was withdrawn from the market. So that is about phase 4 trial. That is ongoing procedure. Now, what are the types of vaccine? Because now, because of the COVID vaccine, we all are going in more depth of this virus, this particular vaccines. So just keeping COVID in mind, you should be knowing that what are types of vaccine. It is in general applicable to all particular vaccinations. Now, it can be a whole viral vaccine, right? It can be inactivated or weakened virus vaccine, which used to form a virus that has been inactivated or weakened so that it doesn't cause a disease, but it still generates the immune response. That is a inactivated, weakened whole cell virus vaccine. Second is protein based, because we know that the whatever virus is an organism, they have particular protein, and that particular protein is acting as an antigen, and antibody against that particular protein are going to be beneficial in any diseases. So protein based vaccine is the second category. Third is a viral vector vaccine. Here what we do, we use a virus that has been genetically engineered so that it can't cause the disease, but it produces the virus protein to safely generate the immune response. So we use specific vector, and we use that virus to make this vaccination. And lastly, nucleic acid vaccine, where we have RNA, DNA vaccines, and also the other vaccine like mRNA vaccines. Now, whole cell virus vaccine, as I said, that inactivated or live attenuated vaccine, definite advantages are a classical time tested strategy for viral vaccination. Production facilities are available. There is, live as well as kill both available. The live vaccine induce the killer T cell responses and also helper T cells responses antibody. So what we need is T cell response as well as the B cell. B cell is the humoral immunity and T cell is cellular immunity. We need a vaccine to generate both the immunity. Now, killed cell vaccine, they do not have a risk of converting it to virulent from the original cause, because live attenuated, you might have the risk of virulence, just what is happening in oral polio vaccine, because we have vaccine derived or vaccine associated poliomyelitis, because it again becomes virulent, neurovirulent, and that causes a disease. So that is less common with not possible with the killed vaccine. Now, disadvantages, as I said, the attenuated forms of pathogen can convert into virulent form. Killed vaccine, they don't generate the specific T cells response, and usually it takes 10 to 15 years for the concept to the production. So it takes long time. So particularly in COVID scenario, this is not going to be good option unless you have very ready-made the substances available. Now, large quantity of virus is required, and live vaccines require extensive additional testing to confirm their safety. Second group, as I said, it was a subunit vaccine. Here, the second generation vaccines are developed to reduce the risk from the live vaccine. So here we may have a specific protein antigen like a tetanus or diphtheria toxoid, or recombinant protein components like a hepatitis B surface antigen. These particular subunit vaccines, they generate a TH as well as antibody response, but they don't develop the killer T cell response. Just like COVID virus, SARS-CoV, we have spike protein. The subunit of that particular is used for the vaccine manufacturing. It is safe and easier to produce, and growing virus and purifying protein on a large scale can take years together. So they require adjuvant to boost the immune response, and particularly SARS-CoV, the full spike protein of this SARS-CoV can cause antibody which has been generated too much. So that small portion of receptor binding domain has to be used. Viral vector vaccine, yes, we can have a replicating virus or a non-replicating. This is technology basically then tested with Ebola vaccine, and that's why because that thing is ready, there is some company which are using this backbone of this vaccine, and they are coming up in the market very fast because that technology already been tested. So this is another virus which has been attenuated, genetically altered to produce a protein similar to required in a antigen. So after viral vaccine, there can be nucleic acid vaccine. As I said, it could be DNA, RNA, or mRNA. So here, what is the thing? The concept of immunizing with DNA, it began promising results in mice 1993. It showed the protective immunity against the influenza. But for decades, these findings have not been translated in similar findings in humans. So that's why it was not been utilized. But now recently, new modification and formulation have improved this particular performance in the humans. And with an expectation that this approach might eventually lead to a first licensed human nucleic acid vaccine, because till now we don't have a single licensed vaccine for humans of that nucleic acid vaccine. Now they are generally safe and can be developed very quickly, and they require a modification to induce a strong immune response, just like DNA and RNA both. Now DNA vaccine, that is a third generation vaccine, it contains DNA that codes for specific proteins from the pathogen, and this DNA is injected into body. It has been taken up by the cells, which usually with the metabolic process synthesizes the protein, which is based on the genetic code of the plasmid. They have taken up, and then that protein contains the region of the amino acid, so it is acting as a foreign protein, and that's why the body generates antibody, and then the whole mechanism works like that. So DNA vaccine, and alternately it can be used encapsulated in the protein to facilitate the cell entry. So if you encapsulate it with the protein, it will go better to the cells, it will be taken up by the cells, and then the whole machinery will work to produce it antibodies. Then plasmid. Plasmid is very important because it is a small extra chromosomal DNA molecule within a cell that is physically separated from chromosomal DNA, and that can replicate independently. And this particular property has been utilized because it can replicate independently. So once we give it to the body, then replication will start and then antigen will be presented and antibody will be formed. So there are vaccines like Zydus vaccine, Zydus COVID vaccine again prepared by Zydus. They are being used by this particular plasmid technology. Yes, mRNA. This is one of the most advanced thing that we are introducing just mRNA sequence, which is coded for the disease specific antigen. So once it is going inside, antigen is recognized by the immune system and that produces the antibody. So RNA vaccine are the faster and cheaper to produce than traditional vaccine. They are safe for the patients as they are not produced using any infectious elements. So there is no infectious element, so it is safe. And this production is laboratory based. So process could be standardized, scaled, and allowed quick response to large outbreak and epidemics. Because now what is required is when you have the vaccine and the demand, particularly when we are talking about COVID vaccine, demand is huge. So we need a rapid turnover and rapid manufacturing with a safe method. So that's why the mRNA RNA vaccine are going to be very useful. So as I said, the safety, because it is not made with the pathogen particles or inactivated pathogen, they are non-infectious. Second thing is, it is very much efficacious. It is generating a reliable immune response and they are very well tolerated by healthy individuals with fewer side effects. So it is safe, efficacious, and as I said, the production is standardized. So we can make a huge production in a very short duration of time. Only thing is, delivering a vaccine effectively to the cell is challenging because this free RNA in the body is quickly been broken down. So we need to have some procedure, a method by which we can make them reach to the cells. Now, whenever we prepare a vaccine, there is one word about excipients. Now, we add something into the vaccine like excipients, like residual manufacturing compounds that may be present in the vaccine. So, alum salt or gel, they are added as an adjuvant to promote an earlier, more potent, and more persistent immune response to the vaccine. So, they allow for the lower vaccine doses. So, because it is adjuvating, they're increasing, promoting that potency, you might require the lower vaccine dose, so that will decrease the side effect. Second thing is antibiotics. They are added to some vaccine to prevent the growth of the bacteria during the production and storage, because we need to curtail that particular bacteria. Also, egg protein is present in influenza and yellow fever vaccine. We should be knowing that is part of the vaccine. That's why people who are allergic to it, we have to take care whenever there is the egg allergy is there. Formaldehyde we use to inactivate bacteria or viruses in the product for toxoid vaccine. And also there are few excipients like monosodium glutamate MSG that was discussed a lot, because there was one more company of chickenpox manufacturing. They were telling that MSG is having a side effect, so our brand is not having MSG like that. But so that is a part of the vaccine. And there is one other 2-phenoxyethanol is used as a stabilizer, because that will make the vaccine to remain unchanged when the vaccine is exposed to heat, light, acidity, or humidity. So these are the part of the vaccine itself. And in general, if you want to produce a vaccine, what you need? First, you need to generate antigen, right? That is being grown either on a primary cells like chicken cell, or continuous cell lines like cultured human cell, and or a bacteria which have been grown in the bioreactors, or a recombinant protein which is there from virus and bacteria, which can be generated in yeast, bacteria, or cell cultures. So first, by any mean, we just prepare the antigen. Then next, antigen is isolated from the cells that are used to generate them. And after that, whatever the antigen is there, the vaccine is formulated by adding adjuvant, stabilizer, and preservative as needed. The most important thing is now we see all combination vaccines. Now they are very difficult to develop and produce, because there is potential incompatibility and interaction among the antigen and other ingredients they are involved. So till now, if we say almost 28 diseases against which WHO has approved vaccines, which is available, and there are few vaccines with more than one vaccine, like polio we have OPV and IPV, meningococcal we have say ACWY separate, a B vaccine is separate. So these kind of things are there, but at least 28 diseases against which WHO has approved a vaccine which is available. Now let us talk about future. What is future? Future vaccines. The vaccines for the disease which is available, but we need a better vaccine. So that is a future vaccine. Vaccines for the disease that is not available, like a COVID vaccine, or there are other vaccines. which a vaccine is not available, but still the manufacturing process or the development is going on. That's a future vaccine. And lastly, there are few newer vaccine delivery for the better results. So that is the future. Now, most needed and most expected future vaccine is COVID-19 vaccine. So we can't get away without discussing the COVID vaccine, because that is going to be our immediate future. So if we are safe, then our future will be safe. So first, we'll be talking about the COVID vaccine. As there are data showing, there are, as I said, a preclinical phase 1, 2, 3, and then approval. So almost more than 200 vaccines uh, candidates are there. Out of that, only 3 are been approved, and that also are approved in a use for limited use. Like we have a Russian vaccine, 2 Russian vaccine, one is Sputnik and another vaccine which has been approved without undergoing proper phase 3 trial. And also there is one Chinese vaccine, Sinovac, that has been approved for the military quota of the China. So these 3 are approved even without getting the full complete phase 3 trial results. But yes, majority of these vaccine which are going to be there in phase 3 trial, and that has been going on, we are going to discuss that in minutes from here. So if you see the list of COVID vaccines, there are so many vaccines, and you must have heard the name very frequently, regularly in the newspaper, in the news. So these are, we will be just touching about the important vaccines. So in general, when you see, there are vaccines where Indian company are having a collaboration with the international brand, like Oxford, like AstraZeneca is having a partnering with Serum Institute in India. One indigenous product of COVID-19 vaccine, which has been produced by ICMR NIV, National Institute of Virology, with Bharat Biotech. There is one more vaccine, a Sputnik vaccine, which is a Russian vaccine, which was discussed like the Putin has given it to his daughter without any phase 3 trial, and that has been now taken up by Dr. Reddy's laboratory, and they will be doing phase 2 and 3 trial in India. And Zydus is manufacturing their own vaccine, as I said, a plasmid mediated vaccine they are trying. There are a few Chinese vaccines which are already been phase 3 trial, like CanSino, which is, I said, that was available in military recruitment. So we are not knowing much. Sinovac and Wuhan virus vaccine. Now, other like Moderna. Moderna is from USA, and that has also done a very good, the development process. They have reached to very far away, and they will be almost coming up with the vaccine in the next year or early first or second quarter. Pfizer and GSK and Sanofi, and also J&J, Johnson & Johnson, they are also coming up with the vaccines. So these are the important vaccines which I think they can see the future or market earlier than other candidate vaccines. Now, as we discussed, there are inactivated vaccines, wholesale vaccines, subunit, vector-borne. So what are the vaccines in COVID vaccine? They are inactivated. Their inactivated wholesale is ICMR, our own vaccine, indigenously produced vaccine, that is Bharat Biotech and Sinovac. Live attenuated, the Serum Institute is working on a Codagenics. There they are going to use this nasal vaccine. It is going to be having a very good future whenever it comes, because it is not going to come immediately, because nasal vaccine, live attenuated, it will be mimicking the natural infection, so it will be best to have. Subunit vaccine, Novavax pre-fusion nanoparticle vaccine and GSK Sanofi are coming with subunit vaccine. Vector-borne disease, there vector-borne vaccines like adenovirus, they have been used by CanSino and Oxford AstraZeneca. Human recombinant adenovector, that is vector used for Sputnik vaccine. A measles vaccine for Themis as well as Serum Institute. Even Zydus is using that. Rabies vector has been used for Jefferson and Bharat Biotech. So these are the vector-borne vaccines. And nucleic acid vaccine, which is going to be a very hopeful vaccine, that is Moderna from USA. RNA vaccine, Pfizer has been working for that, and DNA vaccine, one of the vaccine has been done by the Zydus. So we have a plenty of vaccines which have been in the different stage of development with the different types of vaccine. They are going to come up in the and hit the market. So first is Oxford AstraZeneca Serum Institute vaccine. This is a vaccine which is going to, I think, it can hit the market earliest. That is called Covishield, is the Indian name of that particular brand. They have used a chimpanzee adenovirus vector, which was previously a CHADOX1. Now it has been AZD1222. They are using the spike protein S. They are using a chimpanzee adenovirus vaccine, and single dose in macaque monkeys show protection against pneumonia, but not the infection. So they protected the infection pneumonia. Study in pig shows the boostability in 2 doses, and phase 1 and 2 trial done in UK, and COVID-19 versus this MCV in 1077 subjects. So they have done this particular study, and their subset received 2 doses of 28 days apart, and they gave PCMs prophylactically to all the patients who avoid the side effect of fever, and they were studied for the humoral cellular immunity and safety trial. So after this phase 1 and 2 trial, the results were very encouraging. What they found, almost 91 to 100% subject developed neutralizing antibody. Now mind well, this neutralizing antibody is better to have because there is a strong correlation between the neutralizing antibody and IgG level. See, if you have IgG level that is easily measurable, then you can say yes, reliably neutralizing antibodies are there. So there are few side effects, but they are not much, and they can be manageable with a PCM. There is strong T cell response which is peaked on day 14. Then phase 3 trial, this particular vaccine was done. Already it has been started. It is ongoing, almost 30,000 subjects. And in India also, we know that this particular trial is going on, phase 2 and 3 trials simultaneously. So it was temporarily suspended as we found that there was one case of transverse myelitis in this particular study. But I think still it has been restarted, this particular trial. So you might be getting more results by the end of this year or in the first half of the year. So then comes some mRNA vaccine, that is Moderna, USA made. So this vaccine is encoding S protein sequence that is injected in as a lipid nanoparticle. It is given intramuscular. Here, the S protein is been carried with a nanoparticle. That will take better take up. Phase 1 and phase 2 are been done. Only thing is there is 2 doses are required. So I think all the vaccines are going to be of at least 2 doses, at least 1 month apart or 28 days apart. So here during phase 1 and 2, they tried for different dosages and they tried to safety. Safety was not an issue. And neutralizing antibody was almost in 100% with GMT 2 to 4-fold than the covalescent plasma in almost 100 microgram dose subjects. So it is a good vaccine as far as stage 1 and 2. Phase 3 study, they have enrolled almost 30,000 subjects at 87 sites in USA, and they have used 100 microgram 2 doses 28 days apart. And most importantly, they have two endpoints. One is they want to see the efficacy against the symptomatic COVID-19 and the safety, while secondary endpoint is whether the efficacy against severe COVID or efficacy against infection. So maybe this vaccine may not be working that good, but yes, if it prevents the severe COVID, that is also going to be beneficial. So it may not prevent the infection. So that is a secondary endpoint they studied. Third vaccine which we discussed was Sputnik Russian vaccine. And now Dr. Reddy's laboratory is going to have the phase 2 and 3 study in India. This was invented in Russia, and here they use a modified human R adenovirus vectors. So there are two different vectors. They are usually causing common colds, and what they did, they wanted to give both the same vector, but in phase 2 they give sequential this particular vectors. So on day 1 they gave RAD26 vector, and on day second dose they gave RAD5 vector. So different vector at 21 days, and then they studied. So safety was good, immunogenicity is good. There is a very small study, but yes, they are showing a good result, and they are going for the phase 3 trial, almost 40,000 subject in the 45 medical center. But as we said that Putin has said that it is working, and they have given a permission to use it to the larger scale, but they will be doing the study, and in India, Dr. Reddy's will be doing that study. So what are the disadvantages of this particular fast track? Because it is a human vector, so pre-existing immunity may interfere with the vaccine. That is one thing. Second thing is, if there is a too strong immune response, that may lead to antibody dependent enhancement. So that is also not good. And here, because it has bypassed the phase 3 trial, it may be a risky job if something goes wrong when a vaccine is being given at a bigger scale. And mass vaccination going to go in Russia very soon. Now, these were the important vaccine which I think will come first. There are Chinese vaccine. As I said, CanSino. That is, they are using recombinant adenovirus 5 vector S protein approved for Chinese army. Coronavac Sinovac, that is a company which is manufacturing wholesale formaldehyde inactivated LM adjuvated vaccine, and this is also stable for 3 years, and it is also been finished at almost completing phase 3 trial. These particular two brands, they are having a phase 3 trial in a different country other than China, because what they are saying is now in China they have very almost no cases of COVID. So they are the first one is doing in Saudi Arabia as a phase 3 study, and this coronavirus Sinovac they are doing a phase 3 study in Africa. And last is a wholesale inactivated vaccine by Wuhan. This is also the 3-dose vaccine, 0, 28, and 56 days. And these three are the Chinese vaccine which are almost in phase 3. They are into the phase 3 trials. So there is one vaccine which I think this is because being an Indian vaccine, we'll be just discussing it, Covaxin. This was promised to be, by our Prime Minister has said that it will hit the market by 15th August, but this is the same vaccine. But yes, it has been independently, indigenously prepared by ICMR and Bharat Biotech and National Institute of Virology, Pune. Wholesale inactivated vaccine. The animal study was done in macaque monkeys and it prevented pneumonia and viral clearance. So that's why it is going. It is more chances that it will be working. Neutralizing antibody response also seen, and they have tried with 3 different formulations. So now they will be going for phase 3 trial, and I think 3 or 4 or 6 months down the line, we might be getting the results. This is one innovative technology I am going to discuss, that is pre-fusion protein nanoparticle based vaccine. So here no subunit, no protein, only directly nanoparticles. They have been using that, and phase 1, 2, and 3 trial are going on. So this is Novavax with a different technology, nanoparticle based vaccine. They are going to use it. The most important part is phase 2 study, which was done in South Africa. They utilize this use of HIV positive. So immunocompromised patients, what will be the use of vaccine? That will be that is very important part of the study of this Novavax. So now we are just completed the COVID vaccines. In general, we talked about the other types of vaccine, how the vaccine is been manufactured, and now we'll be looking at the few vaccines for the disease which we have it, but we want a better vaccine. So that's the future vaccine. So hepatitis B vaccine. You must be thinking why hepatitis B vaccine, which is we are having a very nice vaccine, but there is an argument behind it. The vaccine for hepatitis B, it requires 3 doses over a period of 6 months, and studies have shown that 13% of people who receive the first dose of vaccine, they complete the final dose. So rest they stop in between. So that is one thing. And for the adult population, particularly ACAPI has unanimously voted on February 21st that to recommend Heplisav B. This is a vaccine which is available to be used among the individual more than 18 years. And this particular vaccine, it combines the hepatitis B surface antigen with Dynamax property of toll-like receptors, agonists, to enhance the immune response and works like an adjuvant. So that's why it is working faster. It is 0.5 ml dose, which is 25 microgram HBsAg, and with 2 doses series, it given at a 1 month apart, it helps to increase the vaccine compliance. So here with the 2 dose, we can get away, and it shows a significantly high rate of protection, almost 95%. When they studied in parallel with Engerix B, that is 81%, and side effects are comparable. So that is about Heplisav B. That is a 2-dose vaccine which has been approved for adults. Now, why we are discussing hepatitis? Because we know that there are 3 generations of vaccine. First generation, that is plasma derived, second generation, and third generation. Now, hepatitis B vaccine, which has been available for more than 2 decades, but the infection of hepatitis B remains still worldwide because there is a low vaccination rates. So what in 2015, the CDC found that hepatitis B vaccine coverage is only 25% among the 19 years and older, 32% in adults 19 to 49 years, and 17% among the adults. So there is a less vaccination rates, and that's why we are not able to clear this particular infection. Second generation vaccine which is available, they are highly protective, but they are highly protective in young individuals. As the individual age increases, the immune system capacity to elicit a protective response, it decreases, and the likelihood of success decreases further if you consider other comorbidities like diabetes or ancestral disease. So these are the situations where this particular second generation vaccine is not performing well, and that's why the third generation. So what is the difference? See, look at here. Here we have S antigen. So we have utilized the S antigen in that second generation vaccine, and in third generation vaccine we use all these 3 antigens and antibodies against that. So S antigen, PS1 antigen, and PS2 antigen. So this particular new hepatitis B vaccine is called Sci-B-Vac. It is a new hepatitis B vaccine which is safe and has a superior immune response to the comparative vaccine Engerix in phase 3 study. Now, seroprotection rates were almost 99% for this particular Sci-B-Vac, and as compared to Engerix B, which is 95% after completing full 3 dose regime. This particular vaccine was non-inferior following even incomplete 2 doses of the course. So that is something encouraging. And double-blind randomized control, which enrolled almost 2,800 adults, they found that that is a lot-to-lot consistency of this vaccine, and they have applied for regulatory approval in North America, maybe expected later in 2020. So by the end of this year. Now, this particular vaccine is licensed third generation hepatitis B vaccine, which has demonstrated an efficacy of almost 5 lakh patients. They have already been approved and they are commercially available in Israel. So it mimics all three surface antigens of hepatitis B virus. It offers the high level of anti-HBV antibodies. It offers a rapid onset of protection, and it can be administered at lower dose than the competing hepatitis B vaccine and free of next generation adjuvants. So this is about a new hepatitis B vaccine. Now, what about pertussis vaccine? Why we need something better than what we have? Because we know we have a WP vaccine, a whole cell vaccine, and a acellular pertussis vaccine. Now, acellular pertussis vaccine is less effective than expected over the time necessary for protecting humans against bordetella pertussis, because we know there is a waning of immunity, even with AP as well as WP. Even with the infection itself, we get a waning of immunity. So yes, we need a better vaccine. A WP, which is definitely good, but there is a more reactogenicity to that. So AP, which is not preventing transmission of infection, so you need a new vaccine for priming as well as the boosting. So what do we have? We have a new live attenuated pertussis vaccine, which is called BPZE1, in which we have 3 toxins, including pertussis toxin, have been genetically modified and has been developed. Now, this current study, they indicate that upon the nasal administration of this particular vaccine, because it is intranasal to be given, it can prevent the colonization of pertussis disease and ultimately may block the transmission. So that is very important, because this, if you give it to the adolescent population, that will prevent the transmission of the disease. That will, halt the whooping cough in both young children and adults. So if this particular vaccine is successful, the global introduction of BPZE1 could in time eliminate the whooping cough. Now, rabies vaccine. Why do you need a new rabies vaccine? Because we have the vaccine, but it requires a multiple dosages. It requires frequent visits and it's a costly also. So ideally, if you have a single dose to be immunogenic, particularly for pre-exposure prophylaxis, because now we all are having, one thing is we are staying in a society where the plenty of the stray dogs are there, and we are using now pet dogs also in our, we are having a plenty of families having a pet dogs in the family. So new vaccine, what is the candidate? That is a two particular candidates are there. Genetic DNA or RNA vaccine, that is the production and purification relatively easy and cheap. And that's why the immune response are focused on encoded antigen. Unlike viral bacterial vaccine, which also elicits a response against antigen of the carrier. So which limits the usefulness to the homologous booster immunization. So if you use genetic vaccine, there won't be a issue of parallel response to antigen of the carrier. While viral vector vaccine, particularly adenovirus, has been used for this rabies vaccine, they have advantages in the sense that they enter the cell more efficiently upon the binding to the cell surface receptor, and which is as a rule, it renders them more immunogenic. So this is advantage. Simultaneously, the disadvantage is production is very complicated and costly, and they can also only be used only once in individual, as the viral neutralizing antibody induced against the vector backbone will reduce the vaccine update and thereby expression and immunity of the vaccine antigen upon its use for booster. So this cannot be used for the booster. So there are pros and cons about that, and people are working for that. Now, rotavirus vaccine. We know that rotavirus vaccine we are using from 6 weeks onwards. So there is a study, or the efforts are being diverted to see whether we can have some vaccine which can be given right from the birth. So neonatal rotavirus vaccine. So neonatal RV3BB oral live vaccine is developed from a naturally attenuated human strain found in newborns, and it does not cause disease and replicates well in the newborn gut. So there is no issue of maternal antibodies, and even the patient has been breastfeeding. Thus, it has been well adapted into the newborns. So this vaccine was tested in Australia, New Zealand, and now licensed in PT Biopharma in Indonesia, and administered 3 doses starting from 3 to 5 days after birth. So that is the vaccine which is given, neonatal rotavirus vaccine. And this vaccine was found, phase 2 and 3 trial in Indonesia to be well tolerated, better efficacy rate than the same vaccine given in a typical schedule. So plans to have these vaccines available in infants in Indonesian national immunization schedule or program. So that is about neonatal rotavirus vaccine. Now we are thinking about injectable rotavirus vaccine. Why? Because we are giving oral vaccine, and we know that there are many factors which have been affecting that oral vaccine to get absorbed. So we need a vaccine, just like a injectable polio vaccine, getting enthusiasm from that particular vaccine. We have a non-replicating injectable viral vaccine, and they are either killed rotavirus or just subunit vaccine, unlike the current live oral vaccine, which do not require multiplying in the gut to induce the immunity. So we have two things. One is one monovalent parental subunit rotavirus vaccine, which is well tolerated, immunogenic in adult in USA, and in toddlers and infants in South Africa, but it elicited poor response to heterotypic rotavirus strain. So they have monovalent vaccine, but they don't have the cross reaction, the cross protection against the other strain. That's the problem. Second thing is safety and immunogenicity of another parental trivalent vaccine that was tried. So here it is trivalent subunit rotavirus vaccine, which has been accessed double-blind placebo controlled trial in South Africa. The results showed that it is well tolerated with promising P2VP8 IgG and promising neutralizing antibody response across the three vaccines type. So these findings, they support advancing vaccine to efficacy testing against the severe rotavirus vaccine. So there are two vaccines which have been pipeline. One is the neonatal rotavirus vaccine, second is the injectable rotavirus vaccine. Now BCG. Now we know that what is the problem with BCG is BCG's effectiveness low and variable. And there is a waning of immunity also. And we know that BCG protects you from only the CNS meningitis, CNS tuberculosis, and uh, the 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 chest infection particularly. So this particular vaccine is not been used for the primary complex disease. So yes, miliary tuberculosis and CNS tuberculosis only we have protection against this BCG vaccine which is available. Now it is deliverable target for ICMR. ICMR is thought that an efficacious anti-relapse TB vaccine to be there in 5 years and an efficacious TB preventive vaccine in 10 years. This is the deliverable target set by ICMR. Now, if you see, there are three broad groups of BCG vaccine. There, what they are trying. One, what they tried is whatever available vaccine is there, they want to modify in live attenuated vaccine and make it better, because we know that BCG has been developed by multiple passages across. So we have so many serial passages to attenuate that particular bacteria to make it a viral vaccine, so vaccine strain. So what happens that during that process they lose certain antigens, and that's why it is not been effective. So they tried to modify that and use as a substitute to BCG vaccine. Second group is they are using a subunit vaccine, where different proteins, subunit proteins have been used for the vaccine, but here they will be using it post BCG vaccination just to boost whatever the immunogenicity is there given by the BCG. And third group is a nucleic acid vaccine, where again they will be using it for the boosting the BCG vaccine. Now till now the TB vaccine which have been tested at different endpoints with immediate future is a VPM 1002. This is live recombinant vaccine from Serum Institute India. They have already been trying, trials are going on in India. ID93 and GLA-SE, that is a combinant protein vaccine which is discovered by Infectious Disease Research Institute, Seattle, and licensed in Genova Biopharmaceuticals India. And last one is M72 ASO1, that is a recombinant protein vaccine which is by GSK, Belgium, and their phase 2B study has been going on. So that is about BCG vaccine. Now this particular TB vaccine which has been new, it is tried with different mode of delivery other than a conventional one. What we give is intradermal, but now they are trying for intravenous administration also of BCG vaccine, because they think that the BCG vaccine, if it is given intravenous, that it improves the protection from tuberculosis in a non-human primates. That is now 9 out of 10 macaques, they were highly protected against the tuberculosis after IV BCG. So that is intravenous delivery increased the count of persistence of antigen responsive CD4 and CD8 T cells in the lungs. So that has been tried. Second thing is, because the tuberculosis is occurring in the lungs, so the mucosa and the airway mucosa particularly, they are going to be a target. So that's why we are trying for the primary set of disease manifestation tuberculosis lungs. the mucosal route of administration of vaccine. So adjuvant to give the advantage to activating the local natural killer cells. So the second method other than intradermal is sublingual or aerosol trials are going on. And in the mice, these trials are satisfactory, results are showing satisfactory, and hope this future research, especially newborn and infants, can come to our rescue. Now comes the typhoid vaccine. We know that we have the typhoid vaccine. Now, initially we had a polysaccharide vaccine, now we have a typhoid conjugate vaccine. So what are the limitations of currently available vaccines? They are available, they have limitations like they do not provide 100% protection against salmonella typhi, because we have seen the patients already they have given 2 doses of vaccine, still they get a typhoid vaccine, typhoid or enteric fever. Second thing is they do not protect against paratyphoid. So that's why there are efforts being made at new typhoid vaccine, where a recombinant S typhoid outer membrane protein vaccine that is called RSTIV is found to be immunogenic in mice, and it elicits the high serum titers of different immunoglobulin subtypes. And this immunization with the RSTIV also induces a robust CMI, cell-mediated immunity. So that is going to be there. And finally, this mice immunized with this new typhoid vaccine are significantly protected against the salmonella typhi, salmonella paratyphoid challenge with reduced visceral bacterial load. So therefore, there is a potential for this particular vaccine as a novel vaccine candidate for enteric fever, because we are just expecting a vaccine which should give a full coverage of salmonella as well as the paratyphi. Now, what about oral corolla vaccine? I think we have the vaccine Chancol which is available, but it is having, there is a supply gap. So there is research firm Hillman Labs and Hyderabad. They particularly Bharat Biotech are coming up with the phase 3 development of an oral corolla vaccine, Hillcol, which can potentially fill the supply gap. What about group A streptococcus? We have the peptide antigen derived from M protein. They are the most extensively studied vaccine candidates, and all current group A streptococcal vaccine candidates in clinical trials are designed based on a M protein derived protein peptides. So there are multiple vaccines have been there. Various multivalent vaccines are derived from N terminus of group A streptococcal M protein are preclinical and clinical trials, and amongst them these two are in advanced stage, like a 6-valent vaccine and a 26-valent vaccine. Now, what about malaria? I think we have been talking a lot since long about malaria vaccine. So, malaria vaccine development efforts have focused preventing illness from P. falciparum and to a lesser extent against P. vivax, because we considered that P. falciparum is going to be a very deadly disease. More than 30 P. falciparum malaria vaccines candidates are advanced and preclinical and clinical stages of evaluation. And here, the approaches that use the recombinant protein antigen target different stages of parasite life cycle are being developed, because we have the issue in the malaria vaccine development. What? Because the protozoa are very complex organism than bacteria and viruses, and they have complicated structures and lifestyles. So despite the huge number of vaccines which are available, there are none that target the parasitic infection. Second thing is the Plasmodium species have very high rate of replication. So the pharmaceutical treatment are effective at reducing the reproduction rate. They are not halting the reproduction. So that's why there are chances of development of resistance. So whatever vaccines are available, they are giving two main responses. One is the anti-parasitic immunity, and second is anti-toxic immunity. So it has been shown that TNF alpha is having a central role in generating the symptoms experienced in severe falciparum malaria. Thus, a therapeutic vaccine could target the production of TNF alpha that will prevent the respiratory distress and cerebral symptoms, but not the parasitic load. So what we are trying to do is, we are just trying to have effect on a TNF alpha production. To increase the immunity, we need to target multiple phases and in the life cycle of that particular plasmodia. And this is additionally beneficial in reducing the possibility of resistant parasite developing. Now, malaria vaccine, till now only one vaccine which has been approved. We all know that it is RTS-SAS01, which is known by trade name Mosquirix, which is already name suggests the GSK brand. In a phase 3 study, they took two groups, 5 to 17 months and 6 to 12 weeks. And what they found that the 0, 1, 2 and 20 months schedule with and without booster, they studied but the vaccine efficacy is almost very less, around 36%. Without booster, 28% in 5 to 17 months. So in this particular group of 6 to 12 weeks, it is performing even worse than the group of 5 to 17 months. So we have the vaccine RTS-S-AS01, which is a pre-erythrocytic stage hybrid recombinant vaccine based on RTS-S recombinant antigen. And this has been produced by the 2-dose glass vial of RTS powder to be reconstituted with a 2-dose glass vial of AS01 adjuvant system. So when you reconstitute, it will become 1 ml and you have to give 0.5 ml. So you can use it in 2 different patients, but you have to give it intramuscularly. There is no preservative. That's why you can't store it for a long period of time. You have to give it and you have to discard within 6 hours of opening of that particular vial. So what is the current recommendation of WHO? The WHO says at the use 4-dose schedule of this particular vaccine in 3 to 5 distinct epidemiological settings in sub-Saharan Africa at sub-national level covering moderate to high transmission setting. So the published data from phase 3 trial concluded that vaccine has lower efficacy in younger infants. So 5 to 17 months is still somewhat better, maybe 35% of vaccine efficacy. Now hand foot mouth disease. Now you must be wondering why you need a vaccine for that. But we know that this particular HFMD has been caused by the non-polio enterovirus like EV71, Coxsackie, and ecoviruses. So when it has been caused by enterovirus 71, we know that there are high chances of serious complications like myocarditis, aseptic meningitis, encephalitis, and poliomyelitis like paralysis. So we need a vaccine, and we have the vaccine for enterovirus 71. So Coxsackie A16 is the most common cause, but the enterovirus 71 is the second most common cause. There are several strategies, but we have one vaccine which is inactivated enterovirus 71 whole viral candidate vaccine. This was found to be safe and immunogenic in single center study. So there is one more vaccine which got approval in it got approval in December 2015. FDA China approved the first vaccine against EV71, and it has been manufactured or made by the Chinese Academy of Medical Sciences. So this, along with that, in January 2016, there is one more vaccine which is approved, that is EV71 vaccine by Sinovac Biotech. So in China, there are 2 vaccines available for HFMD. One is EV71 and another is again the same but with a different strain. So what we are talking about, Sinovac and this particularly Chinese Academy of Medical Sciences. So they have to be given between 6 to 35 months, and this Chinese Academy of Medical Science vaccine, they have to be given 6 to 71 months. So what is the status at presence of HFMD vaccine? See, multiple circulating strains are there. It may be a Coxsackie virus, enterovirus, so different strains. So very difficult to judge which particular the circulating strain. And there is no cross protection against CA16. So when we have a vaccine of EV71, that doesn't give co-protection from the CA16. And also the concomitant use of other vaccine is not established. So yes, you have to still establish a role in universal immunization program. And inactivated vaccine with weak T cell mediated immune response. It is having a weak T cell mediated immune response. And so what we want, ideally is we should have a live activated vaccine or a bivalent EV71 as well as CA16 to be covered, and it has to be multivalent or maybe a pentavalent vaccine. That should be the future for HFMD. Now there are trial vaccines candidate in RSV. It is called ResVex. It is LM-adjuvated respiratory syncytial virus fusion protein recombinant nanoparticle vaccine, which is investigated for maternal immunization to prevent RSV disease in children, infants. So it is to be given to the mothers. So there are phase 3 trials. It is having a good response. Now one word about hepatitis E vaccine. Now why this is important? Because hepatitis E is one of the important cause of acute viral hepatitis, and it is a non-enveloped vaccine, RNA virus. Total 7 genotypes, but genotype 1 is most common in humans. 3 and 4 can cause infection in animals and can transmit to humans also. This is important because we have to consider China, because their animal usage is too much. Fecal-oral route is transmission, blood transfusion, perinatal vertical transmission is possible in hepatitis E. It primarily affects the young adults and the high fatality among the pregnant women. So it is going to be a good vaccine for particular that group. And whenever we have a co-infection with hepatitis A, because as it is a fecal-oral route transmission, both the viruses, that is going to be dangerous. So there are almost 11 experimental vaccines are been evaluated, and only two vaccines they progress to clinical trials in humans, of which one is Hecolin, that has been developed and manufactured by Chinese brand, and this another experimental vaccine that is progressed to clinical trial that is also not undergone yet the further commercial development. So what is available is only one vaccine, and that is Hecolin hepatitis E vaccine 239 vaccine. So here it is based on 239 amino acid recombinant HEV peptide. It is obtained from open reading frame. That is it encodes the capsid protein, which is very important for hepatitis E, and particularly genotype 1 Chinese HEV strain. So composition is 30 microgram purified HEV antigen, zero point 0.5 ml vaccine, 3 doses to be given, 0, C1 and 6, provides the protection for almost 4 to 5 years. It is approved only in China, and single dose pre-filled syringe is there, only approved more than 16 years. Now this vaccine is readily available in China for hepatitis E. What are the drawbacks? No safety data in pediatric, pregnant women and elderly more than 65. Phase 2 and 3 trials showed protection against type 4 genotype, but not against 1, 2 and 3. So what we are worried about genotype 1 because that is most common for humans. But it has been said that it is demonstrated cross protection against the other genotypes also, even if you find a protection, a direct protection against the genotype 4. Now pneumococcal vaccine. We know we have two vaccines, very nice vaccines, which is PCV10 and PCV13. But what is future? In future, ready-made vaccine has been going to come because Pneumocil, which has been already ready to hit the market, 10-valent pneumococcal vaccine. So Serum Institute is already collaborated with PATH for speedy development of this particular vaccine. And what is the vaccine is, they have replaced few strains and they have used the strain which are more common in Indian setup. So they have substituted few strain from the PCV10 and 13, and they have come up with 10-valent pneumococcal vaccine that is going to hit the market in 3 to 4 months from now. There is one more vaccine, a 20-valent pneumococcal vaccine from the manufacturers who have already manufactured Prevenar. That is 20VPNC candidate that includes 13 serotypes of Prevenar as well as 7 additional serotypes. Because what happens in pneumococcal vaccine, the PCV7 came, and then that 7 particular serotypes that decrease the diseases, but there was serotype replacement. So other serotypes were giving them more diseases. So we went to 10 and 13. Now again, once you give Prevenar 13, there are other serotypes which are coming up. That's why they have added 7 additional serotypes, and that will almost, they say, they will cover up all the serotypes they have been in circulation in USA. So that is going to be coming up in the future. Now, what is the way forward? Because this is a long run, if you say a geographical distribution of predominant serotype changes. So if you try to follow that 7-valent, 10-valent, 13-valent, 20-valent, that is not going to work. So if there is some other approach where it has to be serotype independent, then it will be very useful. So there are growing number of investigational pneumococcal protein-based vaccine that have recently been and are currently being evaluated in clinical trials. So they are protein-based serotype independent subunit vaccine on a combination vaccines and a pneumococcal whole cell vaccine, which is comprised of killed streptococcal pneumoniae organism, which enables the simultaneous presentation of multiple surface protein antigens. Then comes the dengue vaccine. I think what we know is dengue vaccine was available and rather is available, but the issue with that vaccine is you cannot give in a dengue naive patients. The person has to have one episode or has to have suffered from dengue once in the lifetime. So that can be used only in the countries where you have a zero prevalence of that disease, almost 80% by age of 9 years. Because if you give in a naive country, a naive patient, then it is going to give very bad dengue to that particular patient. So that's why it is not, been very successful, but still it is commercially available in few of 11 countries. So there the dose is 0, 6 and 12. Now, so what next? I think Takeda is a company which is going to come up with the vaccine TAC-003, and that has been shown to have a good response based on the 18-month data published Lancet Infectious Disease. This particular Takeda vaccine produced sustained antibody response against all 4 viral strains, regardless of previous dengue exposure or dosing schedule. So it is not required here to have a patient who has already had a dengue in past or they have been vaccinated. So this is one Takeda. There are other vaccines also. They have been Tetravax DV, that is tetravalent admixture of monovalent vaccine. What they did, they prepared a monovalent vaccine for the individual viral strain and they mixed it and they come up with the vaccine. So that is one vaccine, and one more vaccine dengue which is coming up is tetravalent dengue virus purified inactivated vaccine. And this is undergoing a phase 1 trial with GSK and other company. And lastly, a Merck is studying recombinant subunit vaccine expressing Drosophila cells. So these are the future vaccines which are maybe coming up for the dengue. Till now, we have to just face the dengue fever just like that. Now, HIV. Now, HIV vaccine, it has been talked a lot, but there are problems with that particular vaccine delivery because HIV is again a mutable. It is highly mutable. It is a moving target. So HIV isolates are themselves are highly variable. So whatever vaccines are available, the most effective vaccines are whole kill and a live attenuated organism. Now, kill HIV vaccine doesn't retain antigenicity of a live vaccine raises safety issues. So these are the problem. And most vaccines they protect against the disease, not against the infection. So you cannot prevent the HIV infection. Yes, you can protect against AIDS. So HIV infection may remain latent for a long period of time before causing AIDS. So it is very difficult to judge the vaccine efficacy also. Secondly, we don't have the patients who are recovering from AIDS because we know suppose we have COVID patients, they recover and they prepare antibodies. So we can say yeah, these antibodies are going to prevent them from reinfection. While in case of HIV, we don't have any patients who are recovered and then there's no question of reinfection. So it is difficult to measure. And lastly, there is lack of ideal HIV-1 animal model. So that is also a problem with HIV vaccine development. So what are the vaccines which are available or in the development process? There are most initial approaches I have focused on HIV envelope protein. So there are at least 13 different GP120 and GP160 envelope candidates that have been evaluated. So as I said, there are so much of difficulty in manufacturing, but still we don't have any vaccine till now. So in general, we have touched upon the COVID vaccines, we have touched upon the vaccines where we need a better vaccine, and we discuss about certain diseases for which we don't have the vaccines. Now there is evolution in vaccine production. They are using a different technology, and cultured mammalian cells are expected to become increasingly important compared to conventional options such as chicken egg due to greater productivity and low incidence of problem with contamination. Because we know that once we get the chicken egg you use, we have the problem with egg allergy. Now recombinant technology that produces genetically detoxified vaccine is expected to grow in popularity for production of the bacterial vaccine that uses toxoids, because that decreases your chances of getting infection. And combination vaccines are expected to reduce the quantity of antigen they contain and thereby they decrease the undesirable interactions by using pathogen associated molecular patterns. And lastly, one word about a new delivery system. Till now we are using a liquid vaccine. So there is a problem of maybe we have to give injections, we have to use the dosages, and it is sometimes missed. So there are different delivery system has been tried. We know that initially we had an oral typhoid vaccine, but that was available in a capsule form, which was really difficult to digest. But now again we are shifting from liquid to the solid vaccine delivery system. So we are using the tablets and skin patches, even microneedle approaches. They use the pointed projections fabricated into arrays that can create a vaccine delivery pathway to the skin. And also there is a same size patch which is similar to adhesive bandage that contains almost 20,000 microscopic projections per square centimeter. And that increases the effectiveness of vaccination while requiring a less vaccine. So that is about a new delivery system. And one word about edible vaccine. So you must be wondering what is edible vaccine. But here what we use is the plants as a bioreactor for vaccine production. So we get plants we transfer the genetic material to the plants, and then we prepare a transgenic plants, and that once they prepare, and that plants can be used like a tobacco, a potato, a tomato, or banana. They can have genes inserted that can cause them to produce a vaccine usable for the humans. So banana have been utilized or have been developed that produce a human vaccine against hepatitis B. So another example is like expression of fusion protein in alfalfa transgenic plants for the selective detection of the antigen presenting cells. So that has been used for the bovine viral diarrhea vaccine. So this is something new, plants to be used as a bioreactor for vaccine production. That just you should be knowing what is edible vaccine. So lastly, one word about therapeutic vaccine. We have just now talked about vaccine which will prevent the disease, but to treat the disease, there is so much of work going on to prevent the therapeutic vaccine against the infections, allergy, autoimmune disease, addiction, even the smoking. They are trying to develop the vaccine to de-addict the people from smoking. So finally, what is the future of future vaccine? A future is gloomy, or future is COVID vaccines? Future is bright. I think future is definitely bright because the future is you, and that's why you people are going to be a future of all of us. Thank you so much for patiently listening. It is a long topic, yes, I know, but as it was for the people like you who are going to have some benefit in the your exam oriented thing of point of view, because as a practitioner I may not need to know this much of details, but if you just go through this presentation as a whole, you might get many things to be uh, used in your exams. Thank you so much, Dr. Mohit.